Well, hey there, living environment fans. You made it to June. I know you had to take a little day off yesterday to survive to June, but yeah, we made it to June, and now we're really in the hot seat. We're in the final stretch of the racetrack, man. We're headed for home. We're in the home stretch. We're headed to the big finale, which is the Regents exam on June 14th. So we'll do some more Regents review, and best we can do is prep for that test. So let's do that. By the way, it's Friday already. Woohoo! All right. Hey now. Mr. Kovacs class. It's Mr. Kovacs class. You might learn on time. It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class. We're going to go into the nucleus of the cells, and we learned that nucleus is where you have DNA, right? Like the kind of the command center of the cell, and that DNA has all the plans for making proteins. And we learned some specific vocabulary that had to do with DNA in the nucleus, and that was replication, and transcription, both of which happen in the nucleus. Replication is the copying of DNA. Remember, that's where you match A's to T's, C's to G's. A's to T's, C's to G's, right? You unzip the strand, use what's called DNA polymerase, an enzyme. ACE ends in ACE, enzyme. And polymerase matches up A's with T's, G's with C's to make an exact copy of the strand. And that's called replication. Replication happens during interphase when a cell gets the signal that it's time to copy itself and copy its nucleus and go through mitosis. It has to do replication first. Replication. That's different from transcription and translation. The transcription is when you copy just a part, right? A part of the DNA. That part is called the gene, right? Transcription, that uses this enzyme called RNA polymerase. And in this case, we do things just a little bit different, right? We match A's with T's, C's with G's. We match U's with A's. Right, so there's no T in RNA, it's just A, U, G, C. And then that little copy of half a strand basically then leaves the nucleus to go out to a ribosome in order for translation to happen, right? Transcription is when you go from DNA, you split the DNA molecule in half and you copy U's to A's, A's to T's, C's to G's, and G's to C's. Right? U's go, so there's always there's no T in mRNA. And then we call this is copy of the gene, right? This is called the gene template. And it's got every 3 letters of code is called a codon, and those are going to code for a specific amino acid at the ribosome. Transcription happens in the nucleus. In the nucleus, it goes from DNA to mRNA, messenger RNA. And mRNA then leaves the nucleus to get out into the cytoplasm for translation. Transcription, translation. Translation happens at the ribosome in the cytoplasm, right? And that's where tRNA molecules, which have what is called the anticodon, they line up, and if they match the codons on the mRNA molecule, they'll drop off an amino acid. And the amino acids are stuck together through dehydration, synthesis, bonding amino acids together, and we call that a peptide. And as it grows, that gets longer, the chain folds on itself and bends and that kind of stuff, so that you make a protein with the right shape. And I cannot emphasize how much shape is important to proteins. When you are taking a Regents exam, whenever they talk about proteins, make sure you pay attention to the shape. That's what makes them work. And that all happens here when they're made at the ribosome, right? You got messenger RNA, which is the gene, right, the copy of the gene, and then tRNA, transfer RNA, which carries the amino acids to the ribosome for assembly. Translation. Familiar with this chart here? We've used this a few times. This is the chart that we use for translation, trying to figure out if we have the mRNA, like let's say the mRNA said CCU, it tells us what amino acid we dropped off, proline. Okay. Don't remember there's some stop codons, right? UAA, UAGA, UAG. Those are stop codons. That means as soon as they're read, they stop and the protein is finished. Okay, so we've got, and then actually methionine, AUG, is usually the start codon as well. These are, you need to know how to be able to read this chart. Okay, well, we can practice a few right now. And of course, translation is when you put amino acids together. There's 20 amino acids. 20 amino acids. Some are polar, some are not polar, some have different shapes and those kinds of things. So that's why they're not like they're beads on a necklace, but the beads are all different sizes and may attract or repel each other. And that's how you get proteins to fold. And it's the folding of proteins into specific shape that is what's most important about proteins. 20 amino acids. Where do you get the amino acids from? From your diet. If you're like you and me, by eating things that have protein in it. And then in your digestive system, right? Like you chew them with your mouth. In your stomach, pepsin starts to break proteins into peptide chains. Then it goes into your small intestine where trypsin and other pancreatic juice to keep breaking that protein down until the little amino acids where it gets absorbed through the villi of your small intestine and travels through the blood to the cells that need it to make their own proteins. It's like the circle of life. All right, so that's today's Regents Review. Replication, transcription, translation. Replication, transcription, translation. Replication, transcription, translation. Peace.